Welcome to this lecture on the biochemistry of metabolic processes. Today, we're going to explore a fascinating and life-saving metabolic pathway known as ketogenesis. We will understand exactly how our bodies adapt to extreme situations, like starvation, to keep our most vital organs functioning. Let us dive right in and uncover the science behind this incredible biological backup system. To understand ketogenesis, imagine you are driving a hybrid car on a long road trip and you suddenly run out of standard gasoline. Instead of the car completely shutting down and leaving you stranded, the internal computer automatically switches over to a backup battery system to keep the engine running. In the human body, glucose is our standard gasoline, providing quick energy for our brain and muscles. However, during times of fasting, starvation, or very low carbohydrate intake, our glucose reserves run completely dry. To prevent our brain from shutting down, the liver acts like that smart internal computer and switches to a backup fuel system, creating energy packages called ketone bodies. Now that we understand the purpose of this backup system, let us look at how it actually works. Ketogenesis is a highly regulated metabolic pathway where the liver synthesizes ketone bodies from acetyl coenzyme A molecules. This process occurs almost exclusively within the mitochondrial matrix of liver cells, known as hepatocytes. When insulin levels drop and glucagon levels rise, the body breaks down fatty acids through beta oxidation, flooding the liver with acetyl coenzyme A. Because the citric acid cycle cannot process all of this at once, the liver reroutes the excess into the ketogenesis pathway, producing three specific ketone bodies: acetoacetate, beta-hydroxybutyrate, and acetone. Before we look at the chemistry, we must understand why this specific pathway is absolutely critical for human survival. The human brain is a massive energy consumer, but it cannot use fatty acids directly because large fat molecules cannot cross the blood-brain barrier. Without ketogenesis, the body would be forced to break down all of its own muscle tissue to create glucose for the brain, leading to rapid physical deterioration and death. By producing water-soluble ketone bodies that easily cross into the brain, the liver preserves our muscle mass. This is also the biological basis for the popular keto diet and explains the dangerous condition of diabetic ketoacidosis in uncontrolled type 1 diabetes. This biological necessity leads us to the fundamental chemistry of the pathway. Here is the net chemical equation for ketogenesis. Conceptually, this shows how raw materials from fat are converted into usable ketone bodies. The variable A represents a molecule of acetyl coenzyme A, the 2-carbon raw material. W represents a molecule of water consumed in the intermediate steps. On the product side, K stands for acetoacetate, the primary 4-carbon ketone body. C represents the free coenzyme A molecules that are recycled back into the mitochondria. Finally, H+ represents a hydrogen ion, or proton, released into the bloodstream. If this reaction accelerates too much, the massive increase in protons will lower the pH of the blood, causing dangerous metabolic acidosis. Now that we have seen the net equation, let us break down the stoichiometry to see how much raw material we get from a single fat molecule. This formula calculates the yield of acetyl coenzyme A from a fatty acid chain. Conceptually, a long fatty acid is chopped into 2 carbon segments during beta oxidation. The variable N sub A represents the total number of acetyl coenzyme A molecules produced. The variable C subtotal represents the total number of carbon atoms in the original fatty acid chain. By simply dividing the total carbons by 2, we find out exactly how many 2-carbon building blocks the liver has available to start the ketogenesis pathway. If the carbon chain is longer, the yield of raw material increases proportionally. Let us apply this formula to a concrete example. Imagine a person has been fasting for 24 hours, and their body begins breaking down stored fat. We are given a single molecule of palmitic acid, which is a standard 16-carbon saturated fatty acid. We need to find out how many acetyl coenzyme A molecules this will yield. We chose our previous formula because it directly relates carbon chain length to acetyl coenzyme A production. We substitute 16 for the total carbons and divide by 2. This gives us a final answer of 8 molecules of acetyl coenzyme A. Physically, this means 1 molecule of palmitic acid provides 8 individual 2-carbon building blocks ready to be converted into ketone bodies. With our raw materials calculated, we can now determine the actual yield of ketone bodies. This formula shows the relationship between the available acetyl coenzyme A and the final ketone product. Conceptually, it takes exactly 2 molecules of acetyl coenzyme A to synthesize 1 molecule of acetoacetate. The variable N sub K represents the total number of acetoacetate molecules synthesized. The variable N sub A is the pool of acetyl coenzyme A we just calculated. By dividing the available raw material by 2, we find the final ketone yield. If the body breaks down more fat, the pool of acetyl coenzyme A increases, which directly increases the number of ketone bodies released into the bloodstream to feed the brain. Let us continue our previous example to find the final ketone yield from that single molecule of palmitic acid. We are given our previous result of 8 acetyl coenzyme A molecules, and we want to know how many acetoacetate molecules can be made. We use our yield formula because it accounts for the 2-to-1 stoichiometric ratio of the ketogenesis pathway. We substitute our 8 molecules of acetyl coenzyme A into the equation and divide by 2. This gives us a final answer of 4 molecules of acetoacetate. Physically, this demonstrates incredible efficiency. A single molecule of stored fat has been repackaged into 4 distinct water-soluble energy packages that can easily travel through the blood to keep the brain functioning. Now that the liver has produced these ketone bodies, let us calculate the cellular energy the brain can extract from them, starting with the first metabolic step. This formula calculates the ATP energy yield from the oxidation of NADH. Conceptually, when beta-hydroxybutyrate enters a brain cell, it is oxidized back into acetoacetate, releasing high-energy electrons. The variable E sub NADH represents the energy yield in ATP. The number 1 represents the single molecule of NADH produced in this step. The 2.5 represents the ATP generated when 1 NADH enters the electron transport chain. If more ketone bodies are oxidized, this initial energy yield scales up accordingly. Let us calculate this initial energy extraction step for a single molecule of beta-hydroxybutyrate. We are given that the oxidation step produces exactly 1 molecule of NADH, and we need to find the resulting ATP. We use this simple multiplication because each NADH provides a fixed amount of energy in the electron transport chain. We multiply 1 by 2.5. The final answer is 2.5 molecules of ATP. Physically, this electron transfer is a critical first step in reclaiming the stored energy, highlighting how reversible these metabolic pathways can be when tissues desperately need fuel. After that initial oxidation, The resulting acetoacetate is split apart, leading us to our next energy calculation. This formula determines the ATP yield from the acetyl coenzyme A molecules entering the citric acid cycle. Conceptually, the 4-carbon acetoacetate is cleaved into two 2-carbon molecules, which are then completely oxidized. The variable E sub A represents the energy yield from these molecules. The number 2 represents the 2 molecules of acetyl coenzyme A. The number 10 represents the ATP produced by 1 full turn of the citric acid cycle. If the cycle runs faster or processes more molecules, the total energy output from this stage increases dramatically, providing the bulk of the cellular power. Let us calculate the energy generated from this second stage of ketone breakdown. We are given 2 molecules of acetyl coenzyme A, and we need to find the total ATP they produce in the citric acid cycle. We use this formula because it accounts for the energy yield of each cycle turn. We multiply the 2 molecules by the 10 ATP generated per cycle. The final answer is a massive 20 molecules of ATP. Physically, this shows that the vast majority of the energy stored in a ketone body is unlocked when its carbon bonds are fully broken down inside the mitochondria of the brain cells. Finally, we must combine these stages to find the total energy value of a single ketone body. This formula calculates the grand total of ATP produced. Conceptually, it simply adds the energy reclaimed from the initial oxidation step to the energy generated by the citric acid cycle. The variable E sub total represents the total cellular energy extracted. E sub NADH is the energy from the electron transport chain, and E sub A is the energy from the citric acid cycle. By adding these together, we get a complete picture of the energy density. If either of these metabolic stages were impaired, the total energy available to the brain would decrease significantly. Let us perform this final calculation to see exactly how much energy the brain gets from one beta-hydroxybutyrate molecule. We are given the 2.5 ATP from the first step and the 20 ATP from the second step, and we need the grand total. We use this addition formula to combine the yields from both metabolic stages. We add 2.5 and 20 together. The final answer is 22.5 molecules of ATP. Physically, this number represents a tremendous amount of cellular energy packed into a very small water-soluble molecule, perfectly designed to keep the brain running during starvation. This brings us to a crucial realization about why the body relies on this specific pathway. We have just calculated a gross yield of 22.5 ATP. However, activating acetoacetate in the brain actually costs the equivalent of 1 ATP molecule. By subtracting this cost, we prove that the true net yield is exactly 21.5 molecules of ATP per ketone body. When you consider that a single fat molecule produces 4 of these ketones, the total energy delivered to the brain is staggering. This mathematical proof demonstrates exactly why ketone bodies are such an incredibly effective and powerful backup fuel, perfectly bridging the gap between fat storage and brain metabolism. To visualize everything we have discussed, let us look at the complete flowchart of the ketogenesis pathway. At the top, you can see 2 molecules of acetyl coenzyme A entering the pathway. The arrows represent enzymatic reactions. First, the enzyme thiolase joins them to form acetoacetyl coenzyme A. Next, HMG coenzyme A synthase adds another acetyl group to form HMG coenzyme A. Then, HMG coenzyme A lyase cleaves this intermediate, releasing 1 acetyl coenzyme A and producing acetoacetate, our first true ketone body. From here, the pathway branches.
acetoacetate can spontaneously break down into acetone, releasing carbon dioxide, or it can be converted into beta-hydroxybutyrate by consuming NADH. This diagram beautifully illustrates how the liver systematically builds these vital energy packages.
We have covered a lot of ground today, so let us review the key takeaways. First, ketogenesis is a vital metabolic adaptation that allows the human body to survive periods of severe carbohydrate deprivation. Second, the liver acts as a chemical factory, converting excess acetyl coenzyme A from fat breakdown into water-soluble ketone bodies that can cross the blood-brain barrier. Finally, this highly efficient process provides a massive amount of alternative energy for the brain, preventing the dangerous breakdown of essential muscle proteins. You now have a deep understanding of this incredible survival mechanism. Thank you for joining this lecture.